Hi, I'm Jamie Lee and this is a video about protein synthesis. There are two stages of protein synthesis. There is transcription and translation. Within transcription there are three stages: initiation, elongation and termination. DNA is found in the nucleus of a cell and it contains genes that code for proteins which are essential within the body. In the circle, there is the RNA polymerase and helicase. The RNA polymerase signals the start of a specific gene and it binds to the promoter. The helicase then breaks the hydrogen bonds between the base pairs. After the helicase has broken the bonds between the base pairs, it unzips the strands, leaving the sense strand exposed. Then, the single-stranded mRNA moves in between the separated DNA strands. The RNA polymerase moves along the sense strand, giving information to the mRNA, telling it which nucleotides to bring in. Here it can be seen that the mRNA is full and has the complementary bases to those of the DNA. However, thymine is replaced with uracil, which is complementary to adenine. When the RNA polymerase has reached the end of the gene, the mRNA is released from the DNA. Then the hydrogen bonds form between the bases on the sense strand and the antisense strand, which pulls them together. Then the RNA polymerase and the helicase detach themselves from DNA, allowing the two strands of DNA to form the double helix shape. Then we move on to translation, in which there is initiation, elongation and termination. As it can be seen, the mRNA has many bases. However, they are not seen as one long code like this. They are actually in groups of 3 bases, and these are called codons, which code for one amino acid. There will also be a codon to signal the start of a gene and another to signal the end of the gene. During this stage of translation, the mRNA attaches itself to the ribosome at the 5' end. This guy here represents a tRNA molecule. However, they actually have a cloverleaf shape and at the bottom they have an anticodon. The anticodon is complementary to the codon on the mRNA. For example, if the codon was UAC on the mRNA, then the anticodon would be AUG on the tRNA. This is the most important part of the tRNA, as it tells it which amino acid to collect. A tRNA will only collect one type of amino acid. A tRNA molecule with a complementary anticodon to the start codon will move into the ribosome. Then another tRNA moves into the ribosome and attaches to the next codon. And then the rRNA forms a peptide bond between the amino acids. Then the ribosomes move along the mRNA to allow room for another tRNA to move in. The tRNA detaches from the amino acid and moves back into the cytoplasm. Then another bond is formed between the amino acids. This process then repeats until the ribosome gets to the stop codon. This will stop any more tRNA molecules from bonding to the mRNA. At this stage, the amino acid chain is called a polypeptide chain. The ribosome then releases the mRNA strand and the polypeptide chain. The mRNA is then broken down and can be reused in the future. The final stage of protein synthesis is when the polypeptide chain moves from the ribosome to the rough endoplasmic reticulum. Then it is transported to the Golgi body to be packaged and modified before it leaves the cell altogether. And that's about it, so thanks for watching.